Hi everybody. Let us discuss about blood grouping system. In our beginning of 19th century, Carl Einstein also discovered the blood grouping system. So Einstein also showed that the people would be divided into three groups, now called ABO, and inherited characters of aerosol surface detected by specific allo antibody. Blood group antigens may be protein substances, glycoprotein with antibody recognizing primarily the polypeptide backbone. Glycoproteins. Glycoproteins are the antibody recognizing carbohydrate moiety. Glycolipids with the antibody recognizing the carbohydrate portions. The image also depicts blood antigens which was situated on the erythrocytes. Erythrocytes, uh, erythrocytes linked with fucose, galactose, N-acetylglucosamine, N-acetylgalactosamine. So it mainly based on the what are the components joined with erythrocytes. So these bindings also determine the blood grouping. For example, erythrocytes binds with galactose, N-acetylglucosamine, galactose, and N-acetylgalactosamine. So this blood group also known as A blood group. Erythrocytes join together with galactose, N-acetylfucose, galactose, again galactose also linked with fucose and galactose. So this blood group also known as B blood group. Then erythrocytes, the O enzymes inactive. Erythrocytes binds with galactose, N-acetylglucosamine, galactose. So these three components are common for all these blood group. So erythrocytes linked with galactose, N-acetylglucosamine, and again on galactose. Then galactose also linked with fucose. So this is the as usual form of erythrocytes. Then at the end, what are the components binds with the erythrocyte complex? If the complex is N-acetylglucosamine, so that blood group also known as A blood group. In case galactose binds with galactose, blood group B. Then the galactose won't be bind any complex, so the absence of determination. So the blood group also known as O blood group. So ABO blood group system was the first human red cell antigen system to be characterized. The ABO blood group substances are glycopeptides with oligopolysaccharides side chains. So the manifested ABO epitopes of the same specificity as those present on red blood cells of the individuals whom they are detected. ABO specificity is determined by the presence of terminal sugar in an oligosaccharide structures. The terminal sugar or specific blood group A and B, they are also immunogenic. That means so there are immunogen which can be able to induce our immunological properties. Those substances also known as immunogenic. The red cells express neither A, B, and both A and B or neither. So blood group antigen, the blood group of an individual is determined by the presence or absence of antigens A and B on the surface of the red cell membrane. So A and B on the surface of the red cell membrane. RBC of blood group A carry A antigen. RBC of blood group B also carry B antigen. That means the galactose binds with another one galactose. This is the blood group antigen B determination. A determination, so then N-acetyl glucosamine. So N-acetyl glucosamine binds with galactose. So that blood group also determined as A antigen. RBC of B blood group, RBC of blood group AB have both A antigen and B antigen on their surface of RBC. On the other hand, the blood group O cells have neither A or nor B antigens. The blood group are also determined, differentiated by the presence or absence of two distinct isoantibodies in the serum. A individuals have A anti-B antibody. If the persons also have A antigen on their blood, they have anti-B antibodies in their blood. In case the person who have B blood group, so that person also contain anti-A antibodies. O blood group, O blood group, the person also have both A anti-A and anti-B antibodies in their blood serum. So A individuals also have B antibodies, B individuals also have A antibody, O individuals also have both anti-A and anti-B antibodies. Then H antigens. So the red cells of all ABO groups possess a common antigen, the H antigen. H antigen is a glycoprotein and structurally is an L-fucose. It is a precursor for the production of A and B antigens. So glycoproteins and structurally L-fucose. So it is a precursor for the production of A and B antigens. A and B antigens are formed by addition of N-acetyl galactosamine and galactose, respectively, to L-fucose of H antigen. H antigen, due to its universal distribution, is not that important in blood grouping or transformation or transfusion. However, in a rare instances such as in a Bombay blood, both A and B antigens as well as H antigens are absent in the blood, but it contains anti-A and anti-B and anti-H antibodies. So the Bombay blood group rarely occur the Bombay blood group. So both A and B antigen as well as H antigens are absent in that particular individual blood, but it contains anti-A antibodies, anti-B antibodies, and anti-H antibodies in their serum. So this is one type of peculiar Bombay blood group. Then RH blood group system, Philip Levin in 1939 discovered that the serum of most humans who gave birth to infant with hemolytic disease contained an antibody that reacted with the red cells. In 1940, Lance Trainer and Weiner injected blood from the macaques rhesus into rabbit and guinea pigs and discovered that the resulting antibody accumulates rhesus red cells and appear to have the same specificity as the neonatal antibody. The donors whose cells were agglutinated by the antibody to RH cells were termed as RH positive. Those whose cells were not agglutinated were termed RH negative. The terms RH factor and RH antigens are similar and both of them refer to the RhD antigen. Identification of RH incompatibility. RH typing should form part of routine antenatal examination when the woman is always negative and her partner always positive. Fetal complications should be expected. Women with RH incompatible pregnancy should be screened for RH antibodies by the indirect Coombs test at 32 to 34 weeks of pregnancy and at monthly intervals thereafter. So this is the blood group system and antibody system. So then red blood cell types. So the red blood cells, erythrocytes also have some antigen on their surface. That is group A, group B, group A, B, O. So the image also simply depicts the erythrocytes on their surface antigen, O antigen, so the absence of erythrocyte antigen. So B antigen, so the B antigen also have both A and B antigen. So AB antigen, so the blood group AB antigen also have both A and B antigen. So group B also B antigen, A also A antigen. Then A blood group person also possess anti-B antibodies. Then group B person also possess anti-A antibody. Then AB blood group persons also possess without antibodies both A and B. Then blood group O also possess anti-A antibody and anti-B antibody. Okay, so this is also clearly indicate the blood group system. So then blood transfusion. So blood transfusion, blood transfusion is the process of transferring blood or blood products from one person into the circulatory system of another. Blood transfusion can be life-saving in some situation. When the persons require the massive blood, when the person also cause massive blood loss due to trauma. Then sometimes can be used to replace blood loss during the major surgery. Sometimes the severe anemia or thrombocytopenia. Then when the people suffering from hemophilia or sickle cell disease may require frequent blood transfusion. Before blood transfusion, a series of procedures need to be done to establish the proper selection of blood for the patients. Basically, those procedures try to establish ABO RH incompatibility between donor and recipient and to rule out the existence of antibodies in the recipient's serum that could cause hemolysis of the transfused red cells is to test the patient's serum with donor cells. The universal donor ideally the donor and recipient should belong to the same ABO group. Your blood group cells could be transfused to the recipients of any group as they possess neither the A nor the B antigens. Hence, the O group was designated as a universal donor. Universal recipients, due to the absence of isoantibodies in plasma, the AB group persons were designated universal recipients. because AB blood group, absence of both antibodies, A and B antibodies. So this person also considered as a universal recipients. Complications of blood transfusion. Transfusion reaction is the major immunological complication following incompatible blood transfusion. Other transfusion reaction may be caused to do factors other than incompatibility, such as a person be hypersensitivity to some allergens present in the blood. Transmission of infectious agents through blood is the most important complications. These include virus such as human immunodeficiency virus 1 and human immunodeficiency virus 2, hepatitis virus B, hepatitis virus C, and hepatitis virus D. Then and cytomegalovirus. Some bacteria also, such as Treponema pallidum and Leptospira intervans. Uh, Thank you for watching this video. Thank you.